Hi, so my name is Sarah Escobar, and I did my research presentation on fecal indicator bacteria and connectivity. For the background information, you have to know that fecal indicator bacteria includes coliform and E. coli, and it's used to estimate the amount of fecal contamination in a water source. Here's a picture of E. coli on a petri film. And you have to know what connectivity is, and it's basically ions in the water, and it's the ability of the water to support an electrical current. So, fecal indicator bacteria have been linked to different factors such as urbanization in the Journal of the American Water Resources Association. And they said that the more urbanization there was around a creek, the more fecal indicator bacteria there was in the area. And it was also linked to temperature. And the American Society for Microbiology, they said that an increase in temperature affected the amount of fecal indicator bacteria. And it was also linked to turbidity, which is the cloudiness of water. Here's a picture of a sample with really high turbidity, and here's a picture of something with low turbidity. And that study basically said that there were more fecal indicator bacteria in higher levels of turbidity. And it was also linked to the movement of water. So if a water source was highly mobile and there was a lot of flowing in the creek, then there were less fecal indicator bacteria. So this led me to believe there might be a connection between conductivity and fecal indicator bacteria. And my question was, is there a strong correlation between the conductivity of water and the amount of fecal indicator bacteria, which include coliform and E. coli, in Waller Creek? So my hypothesis 1 was that there is a strong or moderate correlation between fecal indicator bacteria and conductivity in the water of Waller Creek, and my hypothesis 2 was that there is a weak correlation between fecal indicator bacteria and conductivity in the water of Waller Creek. So methods included first going out and collecting water. So different student teams were sent out to different sample sites of water in Waller Creek, and here's one of the sample sites, which is called Adams Park. And there were samples taken at these different sample locations, and they were also using YSI to measure connectivity and other factors. And then back in lab, these samples were put onto 3 petri films, and 2 of them were diluted by a PBS solution. And this is a picture of a 1/10 dilution. This is a picture of a 1/2 dilution, and this is a picture of no dilution and just pure Waller Creek water. And after 24 hours of being in an incubator, student teams came back and they counted the colonies on their petri films. And then all information, including the YSI readings and the amount of E. coli and coliform, were put into a spreadsheet. After putting all the important information such as connectivity and the total amount of all fecal indicator bacteria per sample site into a spreadsheet, it was found that there was a linear correlation coefficient of 0 between connectivity and fecal indicator bacteria, which was unexpected. So here is a trend line for connectivity. So yeah, there was a 0 correlation coefficient, which means there's very weak or maybe even no correlation whatsoever between connectivity and fecal indicator bacteria. But if you put the information into a bar graph, then you can maybe see a different pattern, such as a rising connectivity between certain amounts of fecal indicator bacteria. So here, this kind of maybe an outlier, but there is sort of a rise between about 9,000 fecal indicator bacteria colony forming units per 100 milliliters and 17,000 fecal indicator bacteria colony forming units per 100 milliliters. So there may be a pattern there. Also, possible errors included incorrect calculations or maybe different methods of counting between the different groups. The importance of the study is that fecal indicator bacteria is not actually harmful to humans, but it does mean that there's fecal matter in their water, and that's not good. And also, strong correlation between fecal indicator bacteria and conductivity would mean that the water is not clean, because clean water usually means that there is a low level of conductivity, as you can see in this chart right here. Drinking water is pretty low, and then all other water that is not so clean is really high. Future studies for this include eliminating any possible errors that were made in this experiment, such as different methods between different groups. So maybe if the same person was to look at all the different Petri films from all the different sample sites, then there would be a more consistent number of colony forming units per 100 milliliters, or maybe there would be differences in the level of conductivity. Also, that would confirm that hypothesis 1 was disproved, and hypothesis 1, as a reminder, was that there was a high or moderate level of correlation between conductivity and fecal indicating bacteria. Or the question itself could actually go in a different direction, and it could be changed to look for a different pattern between conductivity and fecal indicator bacteria, such as one that would show an increase between certain levels of fecal indicator bacteria, and maybe that would lead to a more interesting discovery. And yes, that's my presentation. Here are my citations. Thank you.